Hello, my name is KAger, and this is for the Microbiome MCB6670 class. I'm presenting on a novel plant growth promoting rhizobacterium that was isolated from healthy apple tree soil by Hansol Kim, Jinsun Kim, Min Kun Soo, and Min Kun Eun, Jim Young Lee, and Jung Suk Lee. And the reason why I picked this article is because from personal experience as a microbiologist, I feel like we have relied very heavily on old school microbiology, which is culturing. And the new age is kind of the microbiome, like sequencing and looking at whole genome sequencing. And I felt like this article did a really good job meshing the old school and the new school together. So I'm going to present on this, and hopefully you enjoy it just as much as I did. So a little bit about the big picture is agriculture in the 21st century has faced a lot of challenges that they haven't faced in the past. One of them being global food security, with an increase in populations and kind of the decrease in our culture. Everyone's not a farmer anymore, and we have gotten this increase in population and population size that food security is a big concern looking into the future. We are also, sustainability is a big issue in agriculture because we are facing new challenges in this that we haven't faced before. Some of the examples I like to talk about, one that's kind of close to home for those of you that are in Florida, are orange trees. For those of you may know or may not know, but you actually can't buy an orange in the state of Florida. Like if you go to the grocery store, most of those oranges that you see in the grocery store are actually from the state of California or other locations, because the orange trees in Florida only go towards the production of juice. And that wasn't the case in the past. And it's because orange trees now have what they're called HLB greening. And it's this disease that they get that basically is a death sentence to an orange grove. If you live in the state of Florida, you can see that all these orange groves, I kind of say they look skeletal, they look sickly. They don't have that big, lush, tree and with all these oranges on it. And you may still see some of them, but those are normally to the young trees. It's as the HLB greening progresses, it kills them and reduces their production of oranges. So this greening disease has actually wiped out quite a lot of farmers' orange groves, and they have been selling them to land developers. And so the orange industry is kind of taking a hit right now because there is no solution to this HLB greening disease. And that kind of is where scientists step in, because they are trying to find the solution to decrease the disease. And that's one of the examples, and I use that one because it's close to home. But there are other crops that have the same issue. Olives is another one in Europe that is also going through the same crisis, and they have a disease that's affecting them, Xylella fastidiosus, and it is wiping out a lot of the olives, and that's a big crop in that industry. So crop management is facing new challenges, and we're now looking at scientists to get involved to help solve these issues. And before, microbiologists would look at it and have a really hard time kind of trying to figure out what the problem was. But microbiome research, looking at the whole soil ribosphere interaction and looking at multiple aspects and sequencing kind of changed the game for looking at crop management, and helps these farmers and help scientists kind of see what's going on and how to resolve the issue in a way that they never were able to beforehand. So knowing some of these issues that we face in crop management and in, our economics and our world atmosphere right now, I really gravitated toward this article because they actually were able to find a really novel discovery that could help, diseased apple trees and crop management in the future. So let's kind of dive in. First, they did, just like everything you start with soil sampling. They collected from an apple orchid located in the Republic of Korea. They did single colonies were cultured and isolated and then preserved in glycerol at -80. And I'm sure this part is very briefly mentioned in the paper, but I also feel like this is probably the more the harder part about this study is going through all that collection and all those samples to kind of get down to the nitty-gritty. But yeah, the rhizosphere microbiome communities were analyzed, and they discovered this novel species, and that they referred to as B3-10T. And the first question you always ask is, well, how do they know this strain from the rhizosphere soil of healthy apple trees was beneficial? So you start out with any plant growth promoting gene study by looking at how that bacterium does in certain situations. So in plant studies, a lot of them use N-Bethamania, and they use it to look at the growth of the shoot, the growth of the root, the weight, and they use the chlorophyll content to see if that bacterium is a plant growth promoting bacterium. And what they did is, so with the control and the B3-10T strain, they could see that there was a difference in the shoot as well as the root. And then they used actual measurements and were able to look at weight, root length, and chlorophyll, and overall the B3-10T strain had a difference over the control. The other thing that I'm just going to briefly go over, because I'm not quite ready to go into a lot of the gene sequences because I have that a little bit later, but they did do a whole genome sequence study, and they did find specific genes in the strain that are known to being plant growth promoting genes. So I just briefly listed them there, so but I'll go more in depth of it later on in the slide. Another test that they did was a cyrophore production test. And what that means is a cyrophore is a molecule that binds and transports iron into microorganisms. And iron is really important for plant growth in uptake and utilization of the iron from the soil into the plant. So this quick and easy test is used. They used it on a control and on the strain. And that orange halo is that transport of iron, that shown transport of iron into the strain, with that strain. So they know that it does have a cyrophore production. The other one is an ACC denaminase production. So what that means is ACC is a precursor to ethylene, which is known to cause disease or promote plant disease in plants. So having an ACC deaminaminase can help degrade the ACC, and that will help protect the plants from environmental stressors because it reduces the amount of ethylene. So here's what the study found with the ACC and then without the ACC in the strain, and there is a statistical significance between the two. So this confirmed by measuring the alpha ketobyruvate production in the presence of ACC. So the strain B310-10T produced more alpha ketobyruvate by degrading ACC. So the one thing I liked about all this is I just didn't use one thing to confirm that was a plant growth promoting bacterium. They didn't just look at the ACC deaminaminase. They looked at multiple things. They looked at the sequence. Did it have known growth promoting genes in it? And then they did studies to kind of see did just by having that gene, does it react a certain way when you're doing a controlled test? So I was actually really happy with the amount of tests they did to confirm that this specific strain did help the growth in the healthy apple trees. And then with the ACC, it also had known things to help reduce disease, to help with healthy apple trees. So they can confirm that this strain is a plant growth promoting bacterium. And then this picture that I've included is a scanning electron image and the transmission electron image of the actual bacterium that they discovered in this rhizosphere soil. So just because they can confirm that this is the bacterium, I feel like, that's great. But then they had to see, okay, is this something that's already been known? Do we already know that this bacteria works? Do we have any other information about this bacterium? And so they actually did two methods. This is more of like the sequencing part. I'm looking at the background. So the first method is extracting the genomic DNA, and then they looked at the 16S rRNA gene specifically, and they amplified it by PCR. And then that PCR product was purified and sequenced from a DNA analyzer. And with this, they are going to be small sequences. So then they have to align those small sequences into a full-length sequence. And they were able to do, so that was one method, is basically PCR with Sanger sequencing to get one full sequence. And just remember, this is only of the 16S ribosomal RNA region. It is not a full-length sequence. The second method is that full-length sequence. So the second method is the extraction of genomic DNA, and then they sequenced it in a PacBio, and then they did a comparison of the whole genome to that Sanger sequencing, and it showed that those regions were more than 99.9% identical. So I feel like with that kind of confirmed, okay, we do know what we're looking at, we do know what we're seeing, they are similar. It's just method one is only of a specific region, where method two is the whole genome sequence. And then they used those and compared them to genomic related reference strains that had been known before to see if it aligned with anything. So with the method one, sequence. So again, remember, this is not the whole sequence. This is just a small region. This is the 16S region that we always talk about with bacteria. And they did a neighbor joining tree. And with this, so you can see in bold the B310-3 where it falls into that phylogenetic tree. And then when they looked at the comparisons, the highest similarity was only 89.5%. And I put an arrow to it so you can kind of see where that falls. A normal bacterial species within, like that's very similar, falls in within a 98.7%. So because it's like such a difference, it's an 89.5% similarity, so it's over a 10% difference, they can tell that this is actually a novel that hadn't been put into any database before. And since that was like the highest similarity, there were others that had 89.4% and 87.8%. And then what they did, you can see in the legend that it is a 0.05 substitutions per nucleotide position. So with this method 1, there is going to be a little bit of a bigger difference because, again, you're having to take small PCR sequences and Sanger sequences and then align them all together. So there could be some differences, even though like they did do the comparison, but that's why that legend is a 0.05 substitution. And then the last one is they used an outgroup, kind of like just shouldn't be similar at all. It should always be an outgroup, and it does show that it was. So method 2 is looking at the whole genome. And I'm going to kind of start at the bottom where you see the legend does show a 0.01 substitution per nucleotide position. Again, that's going to be a little bit more accurate because they looked at the whole genome, where the other one looked at pieces and then had to align it. So this one is a little bit more accurate because this is also not looking at just that 16S region. This is looking at the whole genome sequencing and looking at the difference. So it does say that the B310T had a total length of 7,701,441 base pairs, and that is the chromosome plus the plasmid. And then one thing they noticed in the whole genome sequence is that the GC content, so they kind of realized looking at these that that falls into the family, I'm totally going to butcher this name, the Chitinophagaceae. Sorry, again, don't know Latin, but they know it falls into that family. But looking at the GC content is a normal scientific usage because GC has a 3 strong bond to it versus the AT, which only has 2. So GC content tends to be the normal, what you look at when you're looking in genome sequencings, because you know that those have a really strong bond. And by looking at this, they can see that it had a 34.2% molecular percentage in the B3-10T strain, which was statistically significant than the normal family. Everything in that family normally has, the lowest is a 38.2%, but it could be up to 51.6%. So that was another thing that looking at the whole sequencing, like the whole genome sequence, they could see that the GC content was different than any of the known species already had been previously published. But then they also produced a maximum likelihood tree, which is going to be on the right. And you can kind of see the ones that were in the last tree is also in this tree. And there is a little bit of a difference, but not much. Like the ones that were most similar are still kind of grouped together. The outlier is still in the outlier. It's just that their substitution per nucleotide position is a little bit more structured, and the nodes as percentages are of 1,000 replicate differences. So it's a highly accurate tree. It's just, I think, another way of looking at the other tree. So I do like that they use both of the methods, because they could have just done the 16S region and been done with it. But instead, they did do the step further in their study to be like, no, let's do this next step to see, just to make sure that they are similar, that they're showing us the same thing, and they are basically saying that this B3-10T strain is a novel strain. But they didn't stop there. So I feel like that's the other thing I liked about the study is I kept being like, wow, they did this again, they did this, and I was always surprised that they kept going that extra step. So then knowing that they had like these similar like species close to it, now there was a big difference that 89.5% difference or similarity, so a 10% difference, but they still did the normal biochemical analysis, which is going to be this figure on the right. API kits are a quick and easy way to look at is this oxidase positive, is it catalase positive or negative. It's just, it goes into but the API kits do more than just that. They do different other reactions, biochemical reactions. So they actually tested it with 3 different kits to look at different reactions with those similar strains. And again, their B310T strain had different reactions than the others that were previously known. They also did a fatty acid difference, which is in that image that has like the ABC, and just to see what the fatty acid composition is. And in the bottom, you can see, yes, once again, the fatty acid composition is also different in this novel strain than the previously known strains. All right, so what can we conclude at the end of all these tests? So they were conclusively able to see that this strain B3-10T was a plant growth promoting bacteria that helped those healthy apple trees stay healthy. And they're in the rhizosphere of right by. So rhizosphere is that area in the environment that affects the growth and health of plants. And it is that boundary between the plant roots and the soil. And that's that microbiome interaction, making sure the plants are able to get nutrients. So yes, this strain was able to be confirmed in multiple tests that is a plant growth promoting bacterium. And so now they have this strain, but then they're like, well, is it already known? Did we just do all this work and it was already known? So that's where the sequencing and analysis comes in, and they did 2 different phylogenetic trees, one on that 16S region and one on the whole genome sequence, and they were conclusively able to confirm that they were able to, that this was a novel strain. So what does that mean? So we have this bacterium, we know it's novel, and we know it's a plant growth promoting bacterium that does help apple trees stay healthy. So how do we apply it? And this paper was just published in 2024. So it was published in January 2024. So nothing really has been able to be moved forward with this because this is a new discovery. But the potential of this discovery is pretty amazing, because One, you can think of it as a biocontrol. You know, the next study could be, okay, we know this is a good bacterium that plant should have, that the apple tree should have. So maybe let's try in a disease tree putting this bacterium in and see if it will help reduce disease. So that's like one study you could do. The second study is, okay, we are going to plant this new apple orchard, and we want to make sure that they stay healthy. So putting this bacterium in the soil, like in a greenhouse or in, you know, in a, like, so you can have some type of control, or in the field, but you can use this bacterium to help those new trees stay healthy so you can have, you know, your crop management. The one thing I would love to see is, so we know this is a you know, a positive bacterium that helps the apple trees, but I do wonder if it could actually help produce more apples. So just because it's a healthy tree does not mean that we can say, oh, it's always going to produce X amount of apples. And that would be really helpful in, like, for farmers' stances or like in crop management, because they really care about the yield, like how many apples am I going to get, and what am I going to benefit from this healthy tree, because it's going to cost them to be putting this, you know, biocontrol of bacterium into the field. So they want to get some yield or some increase in revenue. So does it actually produce more apples? Could be like a potential next study to see. The strengths and limitations of the article and future study. So like I was very pleased with how much they put into the analysis to confirm not only that it was a plant growth promoting bacterium, but also that it was a novel species. It didn't just stop at one, and they definitely could have, but they definitely went even further and made sure that before they said that this is novel, this is something we found, that they put the work in. And again, some of the future studies can be on more apple reductions, or can it be used in a biocontrol application, like actually putting this bacterium into a fertilizer onto the field. And then some of the takeaways I got from this is that I really enjoy that they did like old school microbiology with new school microbiology. So putting both the, you know, isolating on cultures and deep freezing, but also the new school of, you know, Sanger sequencing and PacBio, and actually looking at the whole genome sequence. And then they were able to confirm that they actually had some genes that were known as a plant growth promoter. So overall, I was really happy with the paper. Some of the things that we can talk about in our discussion is, one, is there anything you felt that I missed in the paper that you felt was pertinent? Because they did include quite a lot in their paper, but I kind of did try to stick with some of the main points that I felt were really, like, you know, good for the reader. But did you think I missed something? Number two, how did you feel the authors go about answering the questions? Did you feel like they left anything out? Because I did feel like that they answered everything, but there definitely could be something that I didn't think of that you realize that they're like, oh no, they should have done this, they left it out. Question three, were there any confounders? Question four, without microbiome research and advancement, do you think the authors could have found this strain or had these results? Or, like, do you think that they were, if we didn't start looking at microbiome and ribospheres, do you think that they would have still been able to found this? My opinion is I don't think they would have. I think the microbiome research did open up our eyes quite a bit to things that we were missing before, but that's just my opinion. I'd love to hear what you think. Did anything in the results or presentation confuse you? And definitely point it out and we can discuss further. I will say I'm not the best presenter. I'm still trying to figure out how to upload this. So if you feel like there's something that confused you, please let me know. And the last one is, do you think the author's discovery of a novel strain of plant growth promoting bacteria has benefits to areas of research other than apple trees? So I definitely like to hear from you, and I hope you enjoyed this presentation, and I hope you don't mind that I say a lot. I'm sorry, I'll try to be better. But this was very helpful in presenting because I know I'm not a very strong presenter. So I appreciate you taking the time. Thank you so much and hope you have a good day.